Dr. Smith's Nobel Prize, of course, recognizes the foundation of a new field. His work on site-directed mutagenesis has opened up new avenues for the study of biology. It's provided lots of practical help in the way of biotechnology and for advances in medicine. Well, without more ado, I'd like to introduce Dr. Smith, and he's going to talk to us about synthetic DNA and biology.
Thank you very much. This is a very special occasion for me, and I spent the whole of my professional career in Vancouver, and I suppose there's nothing as moving as coming in this room and seeing an audience like this in your home base. On the 13th of October, I received a fax letter from Carl Olof Jakobsson, who is the Secretary General of the Swedish Royal Academy of Sciences, and I read the first part of the letter to you. Dear Professor Smith, hereby I have the honor and pleasure to confirm in writing that the Royal Swedish Academy of Sciences has decided to award the 1963 Nobel Prize in Chemistry for contributions to the development of methods. I better turn these lights down a bit for you. For methods, I'm nervous too, within DNA-based chemistry, with 1/2 to you for your fundamental contributions to the establishment of oligonucleotide-based site-directed mutagenesis and its development for protein studies, and 1/2 to Dr. Carrie B. Mullis, La Jolla, California, for his invention of the polymerase chain reaction, PCR method. On behalf of the Academy, I congratulate you warmly and hope that you will come to Stockholm to receive your prize from the hands of His Majesty the King on December the 10th. According to the statutes of the Nobel Foundation, each laureate is required to give a lecture within 6 months after the day the award was presented to him. The subject of the lecture should be on or associated with the work for which the prize was awarded and will be published in Les Prix Nobel. In accordance with tradition, most laureates give their lectures during the visit to Stockholm in December. This year, the lecture in chemistry will take place at the Royal Swedish Academy of Sciences on December the 8th at 1:30 p.m. Will you kindly let me know the title of your lecture at your earliest convenience, not later than October the 31st. And the title I sent him was Synthetic DNA and Biology. And what I want to do today is, as near as I can, repeat that lecture. The object of my talk today is to describe to you the evolution of the use of small synthetic pieces of DNA as tools in modern molecular genetics. And there are two underlying motives to this story. One is that one often embarks on a program of research heading in one direction and one ends up in a completely different direction. And the second is that discoveries in modern molecular biology generally involve teamwork. And the work I want to describe today is no exception to both these principles. Before I commence my story, I'd like to remind you of a prediction which was made quite a long time ago in 1917 by the winner of the very second Nobel Prize in Chemistry that was awarded. That was Emil Fischer. And he made this remarkable prediction. His prediction of the future role of synthetic organic chemistry is really it's quite dazzling, both in its daring and in its perception. It dazzles because it predated by over 30 years the demonstration of what the chemistry of the genetic material was. That, in other words, the demonstration that genetic material is DNA. And also, of course, it predated by 50 years the elucidation of the double helix, the basic structure of DNA, which makes it possible for us to do what we do. The double helix allows for three things that are important. One is, of course, the faithful replication of DNA from generation to generation, because each strand acts as a template for the production of the other strand. It's also essential for the maintenance of the integrity of the genetic information that that DNA encodes. And just to give you some quantitative feel of the nature of this, you should think about what you are yourself in terms of DNA. Each adult individual consists of approximately 10 million cells. You inherited from each of your parents 1 meter of DNA, which went into the fertilized egg cell, and then that replicated many times over, and so eventually you have those 10 million cells, which will therefore contain about 20,000 million kilometers of DNA. And that DNA, by and large, is a faithful replication of those initial 2 meters of DNA. As adults, most of your cells are not dividing. However, a subset of your cells are, those in your bone marrow, those in your gut. And that cell division is resulting in about the synthesis of about 4 million kilometers of DNA, or in the last minute, each of you synthesized about 100,000 kilometers of DNA, and it was done perfectly. And it's interesting that at the same time, all over your body, there's about the same amount of DNA synthesis going on, that is about 100,000 kilometers a minute, repairing the damage that's occurring to your DNA by virtue of the environment in which you exist. That is the chemistry of the cells and the radiation that is contacting the body, and also, of course, the various other things which can damage DNA. The third reason that the double helix is important is, of course, it provides molecular geneticists with a job. And and I should bring to your, let's see, I've got to think what I was going to say next. Oh yeah. Before I get into how you do that, I should put you in a little bit of a perspective related to the understanding of the relationship between DNA and genetics and proteins. And of course, it all starts in the middle of the last century with 3 quite independent events. One was Charles Darwin's elucidation of evolution. The other was Gregor Mendel with his demonstration of inheritance with experiments with garden peas. And then Friedrich Miescher in his discovery of nuclein, that is DNA. Now, the interest in DNA at that time was not because it was known to be the genetic material. It was of interest because it was the second known organic phosphorus compound. Over the years from that time, enormous amount of work was done in genetics, in the chemistry of DNA, and I obviously can't summarize all that at this time, but the really next and most important event relative to my story is the idea of that one gene encodes one protein, which came from the work of Beadle and Tatum. That then really brought us to the time when the study became a more biochemical study, and the crucial, of course, event that made it a biochemical study was the work of Oswald Avery and MacLeod and McCarty, which demonstrate that the transforming principle was DNA. In other words, genetic information could be transferred from one organism to another by a pure chemical, and that pure chemical was DNA. The other things that happened subsequent to that, which were crucial, were the work of Edwin Chargaff, who, in doing chemical analyses on DNA from a wide variety of species, observed that there was always a constant relationship between the amounts of purines in the DNA and the amount of pyrimidines in the DNA. So there was always an equal amount of adenine and thymine, and there was always an equal amount of guanine and cytosine. And then, of course, the epoch-making event, whose 40th anniversary was just celebrated last year, and that is the elucidation of the double helix as the basic structure for DNA by James Watson and Francis Crick. Another event which is pertinent to Vancouver, and also my story, of course, was the elucidation of the genetic code by Khorana, Nierenberg, and Holley. And this was based on their work, which, using biochemistry, allowed one to decipher the relationship between the sequence of DNA and the sequence of amino acids in proteins. And I'll come back to the genetic code later. The interesting thing is, of course, to mention Gobin Khorana. Gobin Khorana was the man for whom I came to work in Vancouver. in 1956. And again, this is an aside from what I said in my lecture in Stockholm. He didn't win his Nobel Prize till 1968, and that is after he left here and gone down to the University of Wisconsin. But the foundation of the work that led to his Nobel Prize was work that was carried out here in Vancouver between 1952 and 1960. And one of the marvelous gifts, apart from him introducing me, a conventional chemist, to the wonders of biochemistry and the interrelationship with genetics, was a feeling that one could do good work here, because it was very apparent to those of us who were working with Gobin Courant at that time that we were working with a world-class scientist who in all likelihood would win the Nobel Prize. And that always made me know that if I made my career in Vancouver, I could do work that would be as good as anywhere else in the world, and if I didn't do that kind of work, the limitations were not in the environment, were not in the people around me, the students or anything, but were within me personally. And I think that is an important message. To have a mentor like Gobin Courant at the start of your career is a wonderful, inspiring thing. To get back to my lecture, by the way, Courant is the person on the right. What he did was three important things which related to the application of chemistry to genetics. One was he used chemically synthesized pieces of DNA to study the metabolism of DNA by enzymes and RNA. He used synthetic fragments of RNA and DNA to elucidate the genetic code. And he also, subsequently doing the work that elucidated the genetic code, developed the methodology for totally chemically and enzymatically synthesizing genes. And that was important work. Not only did it demonstrate that the gene could be synthesized, but it developed practically all of the basic tools which are the tools of modern molecular genetics. The elucidation of the genetic code was followed by a whole variety of discoveries in the period between the mid-1960s and the mid-1980s. And some of these are listed here. The isolation of bacterial plasmids, that is small chromosomes, isolation of enzymes which allow you to cleave DNA at specific places, the isolation of joining enzymes which allow you to join fragments of DNA together quite precisely, the isolation of the enzymes which synthesize DNA, methods for transformation of a wide variety of organisms, just not bacteria, but plant cells, animal cells, improved methods for synthesis of DNA, rapid methods for DNA sequence analysis, and a whole variety of people who are well known in our field are associated with these discoveries. You can illustrate some of these discoveries quite easily with electron micrographs. This shows the large DNA, which is about 1 millimeter of length, the chromosome of E. coli spilling out of a gently treated E. coli cell. But also it shows, I think that's as focused as you can get it, down here, these small mini chromosomes, plasmids, which are about 1 micron in length and just contain a few thousand base pairs of DNA. The ability of restriction enzymes to cleave these DNAs into specific fragments is illustrated in this picture, and the ability of the joining enzymes to join such fragments together to give intact biologically viable DNA is illustrated here. And lastly, the ability, which is made possible by mild chemical treatment or other treatments of living cells, to insert plasmid DNA back into that living cell and have that DNA handed down from generation to generation of replicating cells is also possible. So those are the basic tools which make the kind of work that I did and many other people do possible. To get back to DNA, the whole basis for both the replication of DNA and the repair of DNA and the chemistry that one can do lies in the base pairing between thymine and adenine and cytosine and guanine. The This is principally a matter of hydrogen bonding between the complementary bases, although there are other elements of structure, non-covalent elements, determine the stability of these structures. And what we set out to do, and again, this was an accident. We'd been working in one area of biochemistry and it hadn't worked out, and Caroline Astel, who is now a professor in the Department of Biochemistry, but then was a graduate student, was in my lab, and we thought we should work on another project. And the question we wanted to ask is, can a short synthetic piece of DNA of defined sequence and length be a useful tool? And there was some background knowledge that you could make affinity chromatography columns, oligothymogenilates attached to cellulose, which could be used to separate by affinity chromatography short lengths of RNA, and also one could isolate naturally occurring tracks of adenylate acid which occur in eukaryote cells. If you're going to use a unique sequence to recognize a unique piece of nucleic acid in a living cell, then of course you're doing something much more specific than that particular experiment did. You want to recognize just one piece of RNA or DNA, depending on your target, and how long the oligonucleotide is, which is going to recognize that, is determined by how big a piece of DNA the organism has. Viruses, which have relatively small amounts of DNA, say 50,000 base pairs approximately, would only need an oligonucleotide that was 9 nucleotides in length to recognize one piece uniquely. On the other hand, the human, with its 3 billion base pairs of DNA in the haploid genome, would require an oligonucleotide of 17 nucleotides. The important thing about this is these lengths should be accessible to chemical synthesis. In other words, you're not setting yourself an impossible task from the statistical point of view. The question is this possible from the chemical point of view? And so what we decided to do, and this is the work that Caroline Astel did, was to make oligonucleotides of defined sequence and length and join them using an appropriate reagent to cellulose to make this kind of affinity matrix, which is insoluble because of the cellulose. And then one could put that in a column and adsorb, for instance, in this case, that we have an oligonucleotide which is TTC, 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 etc., and observe a complement of it, which is GAA, 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 because of the antipolar, antiparallel nature of the duplex structure. And that would form a stable structure at low temperature. And then if you pass buffer through it and it gradually increase the temperature, at some point, these hydrogen bonds would break and this would dissociate, and you could measure the temperature at which this was eluted from this insoluble material. And it's a very simple experiment, but it has a lot of power. And the results of a whole series of experiments of this kind are summarized in the next couple of slides. One is that double helices form very rapidly at low temperature. And this is important because this is in contrast that if you cause the two strands of a double-stranded DNA to fall apart, they will not reform into a double strand at a low temperature, and they have to be annealed at quite a lot higher temperature. Secondly, the stability of duplexes increases with length, although it's a non-linear relationship, and that is the incremental increase in stability gets less and less the longer the duplex gets. And then stable duplexes can be formed with just a limited number of base pairs. And then also GC base pairs tend to be more stable than AT base pairs. Again, other results are as follows. I hope you noticed I'm having my slides alternately yellow and blue for Sweden and white and red for Canada. But the deoxy-T, deoxy-A duplexes are more stable than deoxy-T ribo-A duplexes or ribo-U and deoxy-A duplexes of the same length. If you make a duplex where there's a deliberate match, for instance, a T opposite a T or a C opposite a T or a G opposite a T, that mismatched base pair destabilizes a duplex. It's as if you chopped off two base pairs from the duplex. Whereas the base pair between a deoxy-T and a ribo-G as opposed to a deoxy T and a deoxy G, is only slightly destabilizing. And I want to illustrate this with some simple data. This shows the incremental increase in duplextility with increasing length, and this eventually plateaus over here when this number gets to be 20 to 30. Again, if you deliberately make a mismatch between this T and this T, or this T and this G, you find these duplexes are much less stable. It's as if you've shortened this by 2 base pairs, as I mentioned. These are deoxy with deoxy. If you do one with ribol, again, you find there's an increase in duplex stability. If you put a U opposite a T, then there's a big destabilization, about 2 base pairs. But you notice if you put a ribol G opposite T, there's much less destabilization. So that's a very major difference between the deoxy and ribol series. There are a number of conclusions you can make from these observations. One is that a perfectly base paired short double helix is significantly more stable than a shorter perfectly base paired double helix or a mismatch one. In other words, you should be able to use a perfectly matched duplex of a given length as a highly specific probe. Secondly, the interaction between deoxyribooligonucleotides and DNA is much more specific than interaction of ribooligonucleotides and DNA. And so the take-home message from this, if you want to isolate a piece of genetic material, you're better off searching for DNA with a DNA oligonucleotide probe rather than searching for RNA or searching for DNA with a ribooligonucleotide. So the deoxy-deoxy pairing is the most specific one, and therefore the preferred tool. An oligonucleotide low temperature will form a double helix with a complementary DNA strand in the presence of the second strand of denatured DNA, and that's an important thing for a variety of reasons we'll come back to later on. And then at lower temperature, you'll notice, although if you had a mismatch there was destabilization, there was still a possibility of forming a duplex at lower temperature. And that, again, is important because obviously that relates to the ability to do mutagenesis. Our first idea had been to use an oligonucleotide to isolate a messenger RNA by affinity chromatography when those studies were started in 1968. However, by the early 1970s, when the data, which was very laboriously acquired, had been acquired, then a number of things had happened. And the basic thing that had happened was the whole array of technologies which our genetic engineering had been developed, and so it became much more obvious that one could use a synthetic oligonucleotide as a tool to recognize a specific piece of genetic material, but instead of using it as an affinity chromatographic tool, one should use it as a probe to screen DNA that had been isolated from whatever target organism one was looking for. How do you go about isolating a gene? Well, in principle, if you know the sequence of the protein, you should be able to predict the sequence of the DNA encoding it. There's only a problem here, and that is that the genetic code is degenerate. And that is, there are 64 codons, there's only 20 amino acids, so many of the codons are used more than once to encode the same amino acid. And so you quite often will know the first 2 nucleotides of a triplet code, but then the third one can be different. Occasionally it's unique. For instance, in the case of methionine, there's only 1 codon, and in case of tryptophan, there's only 1 codon, but that is not the general situation. It turned out that a geneticist, Fred Sherman, working in Rochester, New York, had done a tremendous amount of work on the amino acid sequence of mutants of a cytochrome C encoded by yeast. And so he knew not only the wild type amino acid sequence at the N terminus of that protein, he also knew the sequence of a variety of mutants. And amongst the mutants he knew were double frameshift mutants. And these are mutants which are generated by deletion of a base in the DNA at one place and then insertion of a base at another place. And the result of that is, of course, the code is read normally, as you can see here, same amino acids here and here, till you reach the point where there's a deletion, and then suddenly different amino acids start to be incorporated in the protein. And then when you get back to the insertion, then you back in frame again and you get the same amino acid incorporated. it means that if you know this amino acid sequence here, and you know this amino acid sequence here, because of the one nucleotide displacement of the codon in this region, you can put this information together with this information and unambiguously predict what is the sequence of the DNA within this region here. Now, you'll notice that I've underlined a sequence of 13 nucleotides, and that's because this is DNA from yeast. And the calculations that I showed earlier on said that an oligonucleotide of 13 nucleotides should be a unique probe for yeast DNA. And another attractive feature of that was at that time, it was just about all one could do to chemically synthesize or enzymatically synthesize an oligonucleotide of 13 nucleotides. It took about 6 to 9 months' time, and the yields were so low that if you tried to make anything longer than that, you wouldn't have any material left. So this was another attraction about this target. How is the experiment done? And yeast DNA was taken and cut up with restriction enzymes, and we took in this work, and this work was done in collaboration with a group at the University of Washington headed by Ben Hall. They took DNA from the phage lambda. Luckily, that DNA has got a chunk in the middle you can dispense with, and you need the left-hand bit of it and you need the right-hand bit of it for the phage to be infective. You don't need the middle bit, so you can use restriction enzymes to cut the middle bit out and then take the left arm and the right arm and mix it with a large amount of this and a large amount of that with these fragments, and then join them together with a joining enzyme. And what happens is you get tandem repeats. The left arm, a piece of yeast DNA, right arm. Left arm, another piece of yeast DNA, right arm. Left arm, a third piece of yeast DNA, right arm, and so on. And then if you add the appropriate proteins, they will chop this up into viral-sized piece of DNAs and package them. So each of these viruses then has a composite hybrid phage, each of which has a different piece of yeast DNA in it, and this is what's called a library. Now, it's not a very good library because it's a library where all the books are piled in the middle of the floor and you can't find the book you're interested in. However, it can be done, and what you can do is plate these phage out onto a lawn of bacteria, and if you make the solution dilute enough, essentially you get a series of plaques, each of which have been generated by a single phage. What can you do with that? Well, you can then take a replica of it onto nitrocellulose and you can denature the double-stranded DNA. And then you can add radioactive oligonucleotide, and you recollect I said at low temperature an oligonucleotide will form a duplex with denatured double strand of DNA without having the problem of the other strand of DNA competing with it. And so this particularly labeled oligonucleotide fragment should identify any phage plaques that have got the piece of yeast DNA in that has a sequence corresponding to the synthetic sequence, and these are indicated here. These are recognized because of radioactivity, which can be screened for, and then you can use this to go back to this master plate here and actually find out where the phage are that have this piece of DNA, and here you have this there. So that's how the DNA was isolated. Now I'm going to change direction slightly. This shows the life cycle of a bacteriophage that infects E. coli. The interesting thing about this small bacteriophage, first of all, it has a very, very small genome, just over 5,000 base pairs, and it replicates when it's infecting the cell as a double-stranded DNA. But it has the unusual property that when mature phage are being made, it packages just one of those two strands of the double helix in the phage, and so you get a mature phage with a single-stranded DNA, which infects the cell, makes double-stranded DNA, which makes more phage just with single-stranded DNA. And having cloned that gene of the cytochrome C, we wanted to know its sequence. And the reason this phage is important, in the mid-1970s, when that phage was, when the yeast DNA was cloned, the person who was doing the most exciting work in the world on DNA sequence, that is the actual arrangements of bases in DNA, was Fred Sanger in Cambridge in England. And he was working, trying to find out the sequence of this DNA. And I was lucky enough to go and spend a year in his laboratory. and work on the project of working out the sequence of this DNA, which was done, and it was the first genome sequence ever to be done, and also, of course, at the same time, learning his sequence methodology, which was good and great, and it was a wonderful, wonderful year for me. The methodology he developed, not the one we worked on at that time, but the one that's nowadays used, is called the dideoxy method. And it works on the principle that you'd have one strand of DNA as a template, and that's why having single-stranded DNA was important, and a shorter complementary strand as a primer for DNA polymerase. And if you add the 4 normal deoxynucleoside triphosphates, of course, this will get extended using this as a template, and it will make a double-stranded DNA. But if you add to those 4 normal nucleotides an unusual nucleotide which lacks a 3' hydroxyl group, every once in a while, not every time, but every once in a while, that will get incorporated. And because it's got no hydroxyl group on the 3' hydroxyl of this nucleotide, it can't be extended any further, and so it stops. And so a small fraction of these growing chains will stop. And if you happen to have the A one here, there'll be a stop here, there'll be a stop opposite this T here, and there'll be a stop opposite this T here, giving you 3 different length fragments. And if they are then put into a solution and applied to a gel, and an electrophoretic field is applied across here, you'll find that the small fragments will move down here rapidly, and the larger fragments will move down slowly, and they will separate. And so you can see here 1A fragment ending, and then another one, and then another one. And if you do this for all 4 nucleotides, you can essentially get what's called an interrupted ladder pattern, which allows you to decipher the sequence by just going along from shortest to longest and reading off. So you read here, this is, if you put a dideoxy gene, it means the template's got a C. So you see 3 bands here, it means CCC, and then this means a G, and then this means 2 A's, and then this means a T, and another A, and then a G, and then 3 C's, and so forth. You read along here and get the sequence. And that's beautiful, and it works. Now, coming back to Vancouver, we had a problem, 2 problems really. First of all, our DNA was in a large phage, the lambda, and we needed to move it into something smaller to make it more manageable. And this was simply done by moving a piece of yeast DNA out of the phage into a plasmid, the sort of plasmid we saw electron micrograph earlier on. And this could be propagated, and so you can get lots of copy of this. So that gives you lots of copy of a convenient form of DNA. But the problem is it's double-stranded, and the Sanger sequencing method was developed for single-stranded DNA. Now there's an alternate method called the Maximum Gilbert method, which allows you to do double-stranded DNA, and we use that to get the sequence at the end of the gene, the sequence near this end here and the sequence near this end here, but we couldn't go any further. And one day I was sat there puzzling and wondering, gee, how can we get any further? And I remembered the thing that we'd learned some years ago from Carolyn Astell's work, and then subsequently other work by Shirley Gillam, that in fact you could denature a double-stranded DNA and then add an oligonucleotide, and it would form this duplex, even though this was floating around in solution. And of course this then gets you a primer and a template complex. And it so happened we had an oligonucleotide in the freezer that had been made for other purpose, where I saw was complement of a bit of sequence we knew from here. So I thought, well, here's an experiment we can try. And tried it, and it ran the Sanger sequencing method using this combination. And it was one of those wonderful occasions when you have an idea and you try the experiment and it works first time. And it's one of the most exciting experiences in science. And I've been lucky enough to have it happen. And so it's not a good gel by modern standards, but basically it said that experiment worked, and it worked beautifully and gave us the next bit of sequence. And so we were able, using methods we had available then, to make a series. This is the actual first one that was used to do that sequence, that simple structure we had in the freezer, but it worked perfectly and specifically to do that task. So by making another bunch of specific but simple sequences, we're able to walk all the way along the gene and get the total sequence of that gene using this methodology. And this shows the sequence. Now I realize most of you, particularly those at the back, can't see the sequence, but it's a large fragment of DNA from yeast. It encodes the gene for cytochrome C, which is shown here. And there are other things we were able to do, and I just want to mention them again using oligonucleotides. You know where the protein is and you know where the DNA is. You'd like to know where the messenger RNA, and the messenger RNA starts somewhere in front of the protein coding sequence and ends somewhere after the end of it. And we were able to use oligonucleotides, again annealed to messenger RNA, to show that the 3' end of the messenger RNA was exactly at that place and nowhere else. And we're able to show also that unlike the 3' end, which is a unique and specific end, the 5' end of this gene had many, many start places, which is shown here. And this relates to the fact that the promoter, that is the signal that makes messenger RNA in this particular gene, is a horrendously complex structure which is capable of making many separate starts. Presumably most of these can still function to give rise to cytochrome C. So that's the sort of thing one could do. Now, this brings me to the point that the prize was given for, that is for oligonucleotide mutagenesis. And again, I'd like to quote someone else. And this is Joshua Lederberg, who received a Nobel Prize in 1958. And in his Nobel Prize address, he speculated on the fact that the will-o'-the-wisp of genetics was the need for a specific mutagen that would go to a specific gene and change it in a specific way. And he speculated on how it might be done, but he couldn't say how it could be done for the simple reason that in 1958, the genetic engineering technology did not exist. And so he had to think in terms of doing specific mutation on a living cell with the intact genome. Of course, what genetic engineering has allowed one to do is to take a specific gene out of the cell, manipulate it in the test tube, and then put it back into the living cell. And that changes the whole ballgame. And what brought me to the realization that it was an important thing to do stemmed from that work on that bacteriophage FIX174. This is a picture of the genome. It identifies with letters all the proteins it encodes. And the interesting thing about this, in 3 cases, you get overlapping genes. That is, where the same piece of DNA, the same strand of DNA, is encoding 2 completely different proteins. And the reason it's able to do that is, of course, the DNA has a triplet code. And those triplets are read off one after the other, bang, 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 in a row. And of course, if you moved over one nucleoside and then started reading off in triplets, you would read a completely different set of amino acids. And you can, in principle, could move over yet one more step and read a 3rd set. So a triplet code, by its nature, has the potential of encoding 3 different things at the same time. In principle, if you use both strands of the DNA, therefore you could encode 6 different things, but that makes it altogether too complex. But no one even thought before this sequence was done that even 2 overlapping genes was possible. And this is one of the reasons I've always been a very strong advocate of genomic research, that is sequencing the genomes of organisms, because so far every genome that's been sequenced has given you information that neither classical genetics nor biochemistry, which is the study of the proteins that are produced by the genes, had predicted. And this was the first case, and it's carried on to be the situation. Now, most of these genes are required to produce a phage, but the gene E is an interesting gene. Its protein that it produces like a detergent, and it's charged on one end and lipophilic on the other, and it's responsible for the phage being able to break out of the infected bacterial cell to lyse that cell. If you have a mutant within that gene, the phage is produced, but they just accumulate in the bacterial cell without spilling out. And so that was an interesting thing, because the thought was, when you've got overlapping genes, you'd really like a very specific mutagenic method to say, look at one of those genes and not at the other one. And this again then made me think of those observations that Shirley Gillam had made some years ago, where she'd shown that you could form a duplex between a mismatched oligonucleotide and another strand. And of course, if this is one gene in say, FIX174, you can see how easy it is if you could make a mismatched duplex, and you knew this DNA sequence, which of course we did, to form this, and then using DNA polymerase, you could extend all the way around the circle, and then DNA ligase you could join at this place to make a double-stranded molecule with normal nucleotides at all positions except where you put this mismatch in. And if that was used to infect an E. coli cell, then of course both of these strands would give rise to progeny. This strand, the template, would give wild-type progeny phage, but this strand would give mutant phage. And we decided to test the method, and we decided to test by mutating gene E. And the idea here was to change the codon for this particular amino acid to a stop signal by changing this G to an A. Now the interesting thing is it generates a stop signal here, but it doesn't change the code for valine in the overlapping gene D, because valine is encoded for by GTG or GTA, it doesn't matter. And see, that's why it's so nice to have a precise methodology when you can dissect this gene without interfering with the protein produced by this gene. This is the experiment that was done. And again, this is one of these wonderful things when you have an idea. And literally, the idea came from working in Cambridge when I was there and talking over the idea of mutagenesis with Clyde Hutchinson, who's a professor from the University of North Carolina, who was on sabbatical in Cambridge at that time. And I knew a lot about the biology of Phi-X174, which of course I knew nothing about. So we talked about the experiment literally over cups of tea. I came back to Vancouver and said to Shirley Gillam and Pat Janke, here's an experiment that should be done. And Pat went off and started to do the chemical synthesis, and then Shirley did the enzymatic synthesis, and then did the biology, partly in Vancouver and partly in North Carolina. And this demonstrates the experiment. Here we've got a lawn of bacteria, and they've been treated in a way so that even if the phage have a defective gene E, a defective lytic function, they still form a plaque. And so you can see all these are toothpicks marks, and so you put toothpick with phage on in here, and they've all given a plaque. However, if you plate these out on normal coli without this chemical treatment, you find some places you form plaques, but other places, and here there's 4 out of 25, there's no plaque. And yet you know there's phage there because this experiment says there's phage here. And in fact, if you then isolated DNA from these phage and sequenced them, you found that method had made the precise mutant that you'd wanted to make and had that effect. And so that demonstrated that methodology very nicely. So that methodology worked. If it was going to be useful in general, of course, you should be able to apply it to any gene, not just a gene in Phi-X174. And the problem with Phi-X174 is it's an icosahedral phage. It can only package a certain amount of DNA, so you can't insert other bits of DNA into it. But luckily, biology provides you with another vehicle, and the other vehicle is the filamentous phage. Again, it replicates as double-stranded DNA, and you can isolate this double-stranded DNA from infected cells, but it packages single-stranded DNA in the mature virion. And interestingly enough, the packaging is different. It's filamentous, so if you insert a double-stranded piece of DNA in the double strand here, it will make a longer piece of single-stranded DNA, and it will just make a longer phage, and so you can get phages that are twice as long as normal, incorporating this clone DNA. And this is the basis for being able to do the methodology in a general way. And so one can clone a foreign gene into the double-stranded filamentous phage DNA, and it will generate single-stranded DNA as part of the life cycle of the infection by the phage. And then you can do mutagenesis. And again, as before, you'll get ultimately plaques, some of which are formed as a consequence of mutant phage, recombinant phage, and other ones which are formed as a consequence of wild-type recombinant phage. The only difference here is, of course, there's no phenotype, because you've put any old piece of DNA in, you've not interfered with the function of the phage. And so how do you know the difference between these plaques here, which have got wild-type DNA, and these other plaques here, which have got mutant DNA? And again, this comes back to being able to use oligonucleotides as a tool. You recollect that a perfectly matched oligonucleotide duplex is more stable than one with a mismatch. And of course, once a mutagenic oligonucleotide has mutagenized the DNA, it should have the same sequence. And so the mutagenizing oligonucleotide should form a perfect match with mutant DNA, whereas it will form a less than perfect match with wild type. And so if you, again, take a lawn with phage on and anneal a labeled oligonucleotide to it at low temperature, it should anneal to all those plaques. But if you gradually increase the temperature, it should wash off the wild-type DNA, but at the same time form stable duplexes with mutant DNA. And so here you see the difference between low temperature duplexes and high temperature. And these ones, therefore, are the mutant ones. And then if you go back to the master and isolate the DNA, you should, and you do, get mutant DNA from it. And so this is the methodology where, again, a synthetic oligonucleotide could be used as a tool. And the prize was given now because that work was completed. I forgot to mention that the work with the filamentous phase was done by Mark Zola, who was a postdoctoral fellow in my lab in the early 1980s. So this work was completed in about 1982, and yet the prize isn't awarded in 1993. And the reason for that was that other people started to use the methodology, and the fact that over 10 years more and more people used it convinced, I think, the Nobel Committee that this was an important technology. And the principal way in which it was used was in protein engineering, that is, changing genes in a systematic way so that you could change the sequence of the product protein in a systematic way. And this is what is called protein engineering. I just want to give you a couple of examples of protein engineering, which give you an example of what you can do. And they're not work that we've done in my own group, but have been done actually, both of them in a biotechnology, well, one of them in Oregon, by a group in Oregon. And they're working with the protein lysozyme, which is a very studied enzyme. And structurally, it consists of two domains, one here, of mainly helices, and then another domain down here, which is partly a helix and partly beta-pleated sheets, held together by a long helix. And that protein will denature if you heat it. And what was done in this experiment, which was done by Brian Matthews' group, was at different places to change a specific amino acid from whatever was there to a serine. At this point making a serine, and this point making a serine, or in another molecule making a serine here and another one here, or in yet a third molecule making a serine here and a serine here. And these, of course, make, cysteine. I don't know why I said serine. Yeah, I know why I said serine because there's an S there, that's right. Cysteine, excuse me. And first put your mind in gear before opening mouth is a good rule. And cysteine. And of course these cysteines allow you to form a disulfide bridge at either of these places. And in fact, these disulfide bridges stabilize this structure. So one of the applications is to change the generic properties of the protein. This doesn't change the catalytic property of the protein, but it makes it a more stable protein. And there are lots of potential application for that sort of change in biotechnology. A second example is one where one isn't here trying to change the generalized properties of the protein, but the specific ones. And again, this is an enzyme. This is the enzyme cetylysin, and this work was done by Jim Wells at Genentech. And this enzyme is a classic serine proteinase, which has three essential amino acids involved in the catalytic function: an aspartic acid, a histidine, and a serine. And what this experiment was directed at was changing this enzyme so there was no longer a histidine there. And if you remove any of these three residues, the enzyme doesn't work. And this was done using genetic engineering. And so here's the case where this is a normal enzyme, and here you see this histidine. And what was done in the experiment was to replace it with an alanine. That wouldn't work any longer as a normal protein. However, as an enzyme. However, of course, you could postulate this situation where the substrate, and of course proteases hydrolyze proteins which also contain amino acids, where the substrate might have a histidine which would sit in the catalytic site just in the place where the normal enzyme's histidine sat. And in essence, you're reconstructing the catalytic site by using the two critical amino acids from the enzyme and using one of the third critical acid being by part of the substrate. So it's almost a self-destruct situation. And in fact, this worked. And this is what's called substrate-assisted catalysis. The point about it is that here, by genetic engineering, you're making a normal enzyme doing something completely new. And again, that is another use of protein engineering. The third one, of course, is just the basic one that a lot of people use the method for, is just to find out how a protein works at all. And I don't have any slides to illustrate that. But the point I wanted to make, you can use this technology to change general properties or specific properties, and both these are worthwhile things to do. Now, to finish, one of the things that pleases me more than anything is that in the very first paper we wrote on this experimentation, which was the Phi-X work, at the end of it, I wrote this paragraph. And the thing that has pleased me very much is that, in fact, our predictions of what the methodology would be used for are pretty well held up. They have been used extensively for studying the essential features of DNA that are involved in replication. They've been used extensively in defining what are the signals in DNA that say start making RNA and stop making RNA, how it's controlled. And of course, they've also been used, as I just illustrated, for protein structure functional relationships. Now, another interesting thing is we wrote this paper and we sent it to the journal Cell. And the journal turned it down. It said, this is a technological development of no general interest. And so I just tell you that in case any of you had papers turned down by Cell or Nature or Science. It doesn't matter, it's the end of the world. And eventually, of course, we sent it into Cell in 1977, and it was published in the Journal of Biological Chemistry in 1978. And if you do all those things and you get your paper published eventually, you may end up like this. I thought I would just do one other thing before I finish. If you excuse me, I've gone on a little longer than I should have done. But as well as having to give that talk to the Swedish Royal Academy of Sciences on the 8th of December, I was chosen to give on behalf of the recipients of the Chemistry Prize, that is myself and Carrie Mullis, a short speech at the banquet. The prize giving takes place on the 10th of December in the concert hall in Stockholm, and then there's a marvelous, wonderful banquet with 1,300 guests, which takes place in the town hall immediately following it. And towards the end of that banquet, the one person representing each of the 5 prizes which are given in Stockholm, physics, chemistry, physiology of medicine, literature, and economic sciences, gets to give a thank you speech. And I thought, just to end up, I'd read you my thank you speech. Your Majesties, Your Royal Highnesses, ladies and gentlemen. The King and Queen was there, and the Prime Minister, and everybody. So, on behalf of Dr. Carrie Mullis and myself, I would like to express our deep gratitude to the Royal Swedish Academy of Sciences and the Nobel Foundation for the honor that it has bestowed on us today. Dr. Georges Chapek, in his speech of 1992 in acknowledgement of his prize in physics, conjured up with elegant Gallic flair an image involving the number 137, the ancient Nordic gods Odin and Freya, and his work on subatomic particles. Not being French, such imagery is not within my compass. However, others, most specifically Michael Crichton and Steven Spielberg, in the novel and subsequent film Jurassic Park, have conjured with dramatic impact an image of the use of the polymerase chain reaction, PCR, to recreate the dinosaurs. One could imagine a less dramatic reincarnation that is more relevant to this uniquely special occasion. Suppose that one could use PCR to resuscitate Alfred Nobel, and that one could use site-directed mutagenesis to cure the heart disease to which he and his brothers fell victims. What would he, a vigorous 160-year-old, have to say as he contemplated the approaching 100th anniversary of the commencement of the awarding of the Nobel Prizes? I hope that he would have had enormous satisfaction in the honor and prestige that his bequest has brought to his memory, to the Nobel Foundation, to the 2 Swedish academies, to the Karolinska Institute, to the Norwegian Nobel Committee, and to the Swedish and Norwegian people. I hope that he would be both surprised and pleased to see that this year molecular biologists have won the prizes in chemistry and in physiology or medicine, which speaks to the adaptability of the prize selection process in the face of the unpredictable dynamics of scientific discovery. And he would be pleased, I'm sure, by the action of the Bank of Sweden in instituting a new prize in 1968, the Alfred Nobel Memorial Prize in Economic Sciences. I believe that Alfred Nobel, in contemplating this munificent act of the bank and in contemplating what might happen in the next 100 years, his prize was started in 1901, would be concerned with the problems of that next century. And of course, there is one problem that would attract his attention beyond all others. That is the impact of the uncontrolled growth and demands of the human population on the finite capacity of planet Earth. We, Homo sapiens, destroyed the majority of the large mammalian species in North America and Australasia just over 10,000 years ago. We, Homo sapiens, now are destroying the other species that presently exist on this planet at a rate of about 15,000 to 20,000 per year. Given that the current estimate of the total number of species on the planet is about 2 million, this rate, by the end of the next century, will be equivalent in biological effect to the catastrophic events of 65 million years ago that eliminated not only the dinosaurs, but also the ammonid cephalopods, many echinoids, many genera of foraminifera, and of calciferous phytoplankton. The kind of mass extinction that previously in the Earth's history has required 5 million years for recovery, such recovery resulting in a completely different biota that preceded it. I believe that Alfred Nobel, being aware of the unique and enormous impact of his prizes on world thought and opinion, would wish to see a new prize or new prizes instigated to commemorate the 100th anniversary, perhaps related to studies on the control of human population, perhaps on biodiversity, perhaps on the environment, perhaps on sustainability. I thus want to express my deep felt gratitude for the award of the prize in chemistry in the form of a wish on behalf of Alfred Nobel. This wish is intended only as the greatest of compliments to those who value and support the Nobel Prizes because of the enormous power for good that the prizes represent. I hope that the 100th anniversary can be celebrated by the endowment of new prizes that address these problems of the next century, as I think Alfred Nobel would see them if only we could have worked magic with the polymerase chain reaction and with site-directed mutagenesis. Thank you very much.
Well, Mike, on behalf of all of us present today, thanks for just an outstanding lecture, an inspiring lecture, and that really was the reason. To pass on the inspiration from a Nobel Prize winner to all of us. How many of us in our lifetime have an opportunity to hear the science described that resulted in a Nobel Prize by the person who won the Nobel Prize? It's just been an outstanding experience for me. It's a lecture that will remain in my memory throughout my life, and I'm sure that's true for everybody here, Michael. You know, we've seen this afternoon not only the unfolding of the science that led to the prize, the actual experiments, but I think even more important, we've seen the reaction of the scientists as that series of experiments unfolded. The thrill of asking a profound question or a profound series of questions of nature and then actually receiving the answer, and in this case, the answer that you wanted. Can you imagine the excitement in Michael's lab when they saw for the first time that historic gel which we saw on the screen a few moments ago? That must have been, I'm sure, one of the most thrilling experiences of your life as a scientist. And so, Michael, on behalf of all of us at UBC and in this room, I'd like to thank you, not just for the inspiring, outstanding lecture you've just given, but I'd like to thank you for all of your contributions, for the Nobel Prize, for the donation of your prize winnings to schizophrenia research, and to the advancement of women in science, two extremely important causes, and for providing to all of us here such an inspiring, exciting experience today.
Thank you very much.